Hello, AP Biology. Now, we're going to talk about controlling a genome, prokaryotic style. First thing to understand is about one of the methods that prokaryotic cells and even simplistic gene regulation in eukaryotic cells involves something that's called a feedback loop. Sometimes we could call these feedback inhibition loops, depending on the process that's used. But basically, what it boils down to is in a feedback loop, okay, you've got a system where, your products undergo the chemical reaction and you end up with your your reactants, all right, because that's what goes into a chemical reaction is reactants. So your reactants go into a chemical reaction and they come out and you have product, all right? Now, this is a simple enough process, but one of the things you have to understand about chemical reactions, as long as there are reactants, the reaction will happen. That's not good enough for complex living organisms. Sometimes you need to use reactants to a certain amount, other times you need to speed it up, slow it down. And so what we've got is this process called a feedback inhibition loop. So basically, reactants go into a chemical reaction. The chemical reaction happens. Your products come out. Well, it turns out that your products are actually a substance that stops or slows down the chemical reaction. If you think about it, it's almost genius. You make too much of it, it starts to stop the reaction. You have too little, the reaction speeds up again. So the speed of the reaction, the amount the reaction occurs, is entirely dependent upon how much product is available. Now, this sort of style of feedback loop is used regularly in biochemical reactions. All right. So, yeah, we've talked about that. Sometimes you can also have the presence of a certain substance that promotes a reaction. An example we're going to talk about today involves lactose in bacteria. So if there's no lactose present, the bacteria do not want to waste time or energy producing the enzyme that breaks down lactose. So that section of their genes is only turned on if lactose is present. And that can happen in a lot of different systems as well. All right. Uh, there's a lot of variations to this process, but the main thing is what we just went over. You have a system where the product inhibits the reaction, all right? And that's a very important concept. Now, to really get into how this concept works, we need to talk a little bit about how gene sequences are organized, all right? So, first off, your gene is always divided into several sections, okay? Um, First off, you've got your promoter, all right? Now, the promoter is the sequence of the DNA that is in front of the coding structural gene, and this right here is the structural sequence of the gene. So it is in this part that I'm highlighting right now where you find the structure of the polypeptide sequence, and therefore within this part, the structure of the polypeptide sequence leads to the function of the polypeptide, okay? Now, over here, this part that we talked about first, is a sequence of DNA that actually starts the process of transcription. All right, this is where RNA polymerase binds. This is where the signal for RNA polymerase to begin acting happens. All right. Now, between the promoter and the actual structural sequence is this section right here. All right, this section right here. It's called the operator. All right. Now, the operator is actually, usually it's a sequence that's inside the promoter. So this whole region over here is known as the promoter. But the operator actually is a section where inhibitors can bind and stop the process of transcription. And so the promoter is what causes it to begin. The structural sequence is where the actual key polypeptide structure is located. And the operator is something that we can use to stop the process of transcription. Turn it off or even turn it on. Okay? Oops, forgot something. The operator and promoter together are known as the operon. All right, that's where that comes from. All right. Sometimes the way this functional works with the operator is that you have proteins that repress the substance. All right. So what will happen is a protein will bind to the operator, and it will stop RNA polymerase from transcribing whatever gene it is that we need. Now, these repressor proteins are coded for by other sections of your DNA. All right. It gets complex. That's why you have tons of DNA. But these repressor proteins are coded by other sections of your DNA, and they just kind of happen. They happen slowly, and they happen continuously. So there's always an appropriate amount of repressor protein in a system. All right. Now, repressor proteins tend to be what we call allosteric. All right. Now, allosteric is something that changes shape upon the binding of another substance. So there's two possible things that could happen here. All right. First, you could actually have a repressor protein that is constantly bound to the operator. And then some other substance comes along, binds to the repressor protein. The repressor protein changes shape. If it changes shape, it changes structure. Structure equals function. It can no longer bind to the operator, and then that gene is transcribed. All right? Or you can have a system where the repressor protein just kind of floats around, and then a substance shows up, binds to it, changes its shape, and then it binds to the to the operator, and then shuts down the production. Okay? So there's two things where allosteric binding of another substance can actually cause the repressor protein to stop turning the gene off, or allosteric binding of another substance can actually cause the repressor protein to then attach to the operator and shut the gene off. It can work either way. But the important thing here is an allosteric repressor protein, when something else binds to it, changes its shape, it either becomes inactive or it changes its shape and becomes active. All right, and that's what this diagram over here is all about. It's describing for you how allosteric binding in an enzyme could turn it on or off, which is similar to this. So let's talk about two different examples of the operon. All right, now the first one is what's known as the trip operon. Okay, now trip operon, there is a substance that's a sequence of DNA that's regulating the production of tryptophan. Okay, and what basically happens is when there's a lot of tryptophan, it binds to a repressor protein, attaches to the operator, and shuts down the production. When the levels of tryptophan drop, it ends up releasing because it's getting used, and then the production continues, and then the levels of tryptophan go up, and then the process repeats. So let's take a look at this over here, right? So here you have an inactive repressor, okay? And here you have your gene sequence. Right here is your operator, all right? Now, what happens with the operator is that as tryptophan is produced, all right, so as the amount of tryptophan goes up, it ends up binding to these repressors. These repressors then attach to the operator, and the amount of tryptophan goes down. Well, when it goes down, the tryptophan releases from the repressors, and then it starts making tryptophan again and goes up. And then as that goes up, it binds to repressors, attaches to the operator, tryptophan is no longer produced, the amount goes down. And then when it goes down, the repressor proteins and the tryptophan that's bound to them release, it starts producing more, tryptophan goes up. This is a trip operon. This is actually what we call a repressible operon. Repressible means the repressor protein is inactive at the start. Repressible means that the process is only turned off when the amount of our product gets too high. Okay? Next operon example is what's known as the lac operon. All right, now it's called lac because it's all about lactose. Certain bacteria contain a gene called the lac operon, which can be used to digest the sugar lactose. However, the sugar lactose is not as common as sugars such as glucose, fructose, okay? And so that gene is not always turned on. Why waste the energy producing a protein that you will not use? All right. So what happens is, in this example, the lac repressor is always active. Okay. And you can see this right up here. Here is the promoter, this red section. Okay. Or I'm sorry, this is the operator. Now, see how the operator is being drawn with that specific structure, where the repressor always attaches to the operator. So this is always shut off. Okay? So in this case, the repressor is always active. So inside lactose, and you'll find this as we move forward and talk about organic macromolecules, there are structures called allolactose. They are basically an isomer of lactose. Similar chemical structures, same chemical formula, slightly different structure. Okay, and it's always present in a certain percentage. That's just the way nature works. Sometimes things are assembled slightly different. Biggest example you'll know if you go by rubbing alcohol. Sometimes it'll just be called propanol, all right, or it'll be called isopropyl alcohol. Same exact chemical formula, slightly different structure. Structure equals function, slightly different chemical activity. Same thing here. Allolactose is an isomer of lactose that is always present in small amounts in lactose. Well, since it's not going to be digested, this is what's brilliant about this. The allolactose, which is represented right here by this purple structure, binds to the repressor. What this does, and you can see it right here, it inactivates the repressor. The repressor no longer works. Well, if the repressor doesn't work, it never binds to the operator. And if it never binds to the operator, lactase is made. And lactase is the enzyme that allows bacteria to digest lactose. So what you've got here is what we call an inducible operon. Okay? So before, as the substance that we were generating built up, the operon turned off the transcription of the gene. Here, the transcription of the gene is always off. And then the presence of a certain substance, in this case lactose, and specifically its isomer allolactose, stops repressing the gene. And then all of a sudden, the gene is turned on, the substance the gene makes, in this case lactase, is generated. Lactase destroys the lactose, and as lactase destroys the lactose, when there's no more lactose left, the repressor binds again, and then the substance shuts off. All right? Feedback loops, very important process in biochemical reactions, specifically in prokaryotic gene regulation.